All living organisms are composed of cells. As we learned previously, a cell is the smallest unit of life that can function independently. And we can see two examples of single-celled life forms on the screen in front of you. Before we talk more about cells, though, we need to familiarize ourselves with the metric system, because in biology, we use the metric system when we are talking about the relative size and or shape of different types of cells. So the metric system uses meters to represent length, and meters are a lowercase m. Liter is for volume, and that's a lowercase l. Gram is a measure of mass or weight, and that is represented by a lowercase g. And Celsius is for temperature, and that's represented by that little circle with an uppercase C, and that stands for degrees Celsius. Now, in addition, we need to learn some prefixes. By adding these root words to the beginning of the word meter, for example, it can tell us the relative size and scale. So a kilometer, which is really pronounced kilometer, is 1,000 meters. A meter is a meter, of course. Now, what if we put centi in front of the word meter? A centimeter is 1/100th of a meter, so that's pretty small. A millimeter is 1/1000th of a meter, so that's very, very small. But when we're talking about most cells, we need to go even smaller and use micrometers. Micrometers are 1/1,000,000th of a meter, and most cells are in the range of 10 to 100 micrometers. Without a microscope, we are not able to see most cells with the human eye. Looking at this figure on your screen, you can see a variety of different things, including a molecule, a virus. Viruses, by the way, are not living things. Bacteria, plant and animal cells, and so on and so forth. Take a look at this website when you're at home and you have a second, because it's a really nice tutorial that shows you the relative size and scale of different objects. So before we talk any more about cells, let's talk a little bit about the history of how microscopes were first used. And the fancy word for using microscopes is microscopy, which is the title of the slide on your screen. So Robert Hooke is credited as being the first scientist to describe cells, and he had a very primitive light microscope. Of course, in the 1600s there wasn't electricity, so the light source was either sunlight or candlelight, and he was a botanist, so he was actually looking at thin slices of cork tissue. So cork comes from the bark of a type of oak tree. Then about 150 years later, cell theory was finally hypothesized. And cell theory is composed of two parts. Number one, all living things are composed of cells. And number two, all cells are formed from previously existing cells. Next, we're going to learn about some of the types of microscopes that we can use to look at cells. The first is called a transmission electron microscope. It's a very powerful tool that allows us to see the internal structures inside a cell in unprecedented detail. A scanning electron microscope is also very powerful. However, this allows us to see the details on the surface of a cell. So it shows that cell in 3 dimensions with all of the parts on the outside of the cell. And finally, light microscopes are less powerful, but they're very useful for biologists. This allows us to generate color images of living cells. So you might be asking yourself, why is it that cells are so small? Well, the short answer is, smaller cells are able to communicate more efficiently with their outside environment. And the reason for this is because smaller cells have more surface area that is relative to their volume. And this high surface area allows these cells to quickly exchange materials with the surroundings around them. So here's a short list of 6 features that all cells have in common. So this includes both prokaryotic cells and eukaryotic cells. Number 1 is DNA. Number 2 is RNA. Number 3 is ribosomes. Number 4 is proteins. Number 5 is cytosol. And number 6 is cell membrane. And we're going to define all of these terms throughout the lecture, but I do want to mention cytosol. Cytosol is that fluid that we find inside a cell. And we're going to learn another word for it, is also called cytoplasm, but cytoplasm technically involves organelles as well. Cytosol simply means that fluid, and that's important because remember, there are no organelles in prokaryotic cells. Here's our first question. Which cell shape has the highest ratio of surface area to volume? So we can visually see these different cell shapes, and the first one is a circle, the second one is a rectangle, the third one is kind of a blob, and the fourth one is a blob, but it's extremely flattened out like a pancake. Which one has the highest ratio of surface area to volume? Pause the lecture now if you want to wait to think about it. The last one is the correct answer. It's the cell that has lots of little finger-like projections coming out of it. It has a much higher surface area relative to the internal volume. Prokaryotes, as we learned previously in Chapter 1, are the most ancient forms of life. Remember that all prokaryotic cells lack a nucleus. They still have DNA, but the DNA is not contained in a nucleus organelle. Eukaryotes have cells with a nucleus as well as other membrane-bound organelles. In this table, it allows us to compare prokaryotic versus eukaryotic cells. And we need to understand just a few parts of this table. For example, nucleus. In bacteria and archaea, the nucleus is absent. In eukarya, it's present. Membrane-bounded organelles are absent in the bacteria and the archaea, but they are present in the eukarya. And then I want you to know typical size. So this far column. Bacteria and archaea are on average only 1 to 10 micrometers. So that's super small. The domain eukarya are 10 to 100 times larger. So they're 10 to 100 micrometers in size. Here's that picture of a generalized bacteria, and it's important to note that there are hundreds, if not thousands, of bacterial forms. This just happens to be a generic model of one. But in reality, if you went on to take a cell biology class or a microbiology class, you would learn a huge diversity of what bacteria look like. Now we can start discussing the eukaryotic cells. Remember that animals are eukaryotic organisms because they have eukaryotic cells. They have membrane-bounded organelles, and you can see on the screen there's a huge diversity of organelles which we're going to address one by one in the upcoming slides. Plants are also eukaryotic, but notice the cell wall and the chloroplasts. So animal cells did not have these features. In addition, because there is a nice, dense cell wall around the cell, it needs to have little tunnels that allows it to communicate through that cell wall. So sure enough, plants have to have plasmodesmata that allow for communication and the transport of molecules through that cell wall. Here's our next challenge question. How many of these features does a typical bacterial cell have? And your choices are DNA, cell wall, nucleus, ribosomes, and/or cell membrane. Pause the lecture now if you don't want to hear the answer. And the answer is 4. Bacteria have all of the above except for a nucleus. When you're studying for the exam, make certain you can distinguish prokaryotic versus eukaryotic cells. All right, so now we can look at the specific types of organelles that we find in a typical eukaryotic cell. In our tour of the cell, we're going to organize it as follows, starting with number 1, border control. The cell border has a plasma membrane. A membrane surrounds each and every cell, and these cell membranes are composed of special molecules called phospholipids. Now remember, we've already talked about this term lipid, right? Lipids are things such as fats, but steroids are also included in this group. And remember, lipids are not soluble in water. This is a very important property of these phospholipids because it's going to help separate the cell contents from the watery environment around it. The phospholipid is this little circle structure with tails. The hydrophilic head is that circle structure, and it forms polar bonds which are attracted to water. Remember, hydro means water, and philic means loving. The hydrophobic tails are seen below the head, and these have nonpolar bonds, which means that it repels water. This is the reason that we're able to separate the watery outside of a cell from the watery parts inside a cell. Because of this chemical structure, phospholipids spontaneously form bilayers when we place them in water. And this is very important because these bilayers can serve as membranes that surround each and every cell. Besides phospholipids, cell membranes also contain proteins, and the proteins are usually represented as these big purple blobs. So here we can see large proteins embedded in this cell membrane. There are several different functions of these proteins, including transportation, enzymes, recognition, adhesion, and reception of different types of molecules. All right, here's our next question. Cholesterol is a molecule in animal cell membranes. Since cholesterol is hydrophobic, where is it most likely to occur? Is it found in the head region or the tail region of our phospholipid bilayer? Pause the lecture if you don't want to hear the answer. It's B, which is that region in the tail part of the phospholipid bilayer. Because remember, the tail region is where we find the hydrophobic zone. When we're studying for the exam, make certain that you understand the chemical structure of phospholipids and how it enables them to form a bilayer in watery environments. All right, let's move on to number 2, the genetic control of the cell. We can think about this as being the brain of our cell, the nucleus, and the ribosomes. What is the structure and function of the nucleus? Well, number 1, it contains DNA. Specifically, it contains the chromosomes which hold genes. And notice my shorthand. After the word chromosomes, I write C apostrophe and then somes. That is my shorthand for the word chromosomes. I like to teach you ways to take shorthand so that you can more effectively take notes while you're listening to my lecture. The nucleus is surrounded by a membrane called the nuclear envelope, and the nuclear envelope has little holes or pores in it that regulate transportation, and these pores are called the nuclear pores. In the center portion of the nucleus is a region called the nucleolus, and this plays a specific role in assembling the ribosomes. Notice in this diagram, the ribosomes are the little dots that are found on the surfaces of these membranes. Ribosomes are also an important part of the genetic control of a cell, largely because ribosomes play an active role in synthesizing proteins. And remember, proteins give us traits. These proteins are essential for living things to exist. Ribosomes carry out protein synthesis by translating information that is carried on the messenger RNA. Remember, we learned about RNA in the last lecture. It's a type of nucleic acid. Now, the shorthand for the word messenger RNA is written as follows. It's lowercase m and then uppercase R, N, A. Where do we find ribosomes? Well, we find them free, floating around through the cytosol in the region we would call the cytoplasm, or they get attached to membranes, including attached to the endoplasmic reticulum. And notice there's a shorthand for the word endoplasmic reticulum as well, and that is uppercase E, uppercase R. Prokaryotic ribosomes differ from eukaryotic ribosomes. Remember that both prokaryotes and eukaryotes possess ribosomes, because without ribosomes you can't make proteins, and without proteins you can't be a living thing. This is one of the ways that antibiotics function. There are some types of antibiotics that target prokaryotic ribosomes. So if you're a bacteria and you can no longer synthesize proteins, you're going to die. How is it that DNA controls the cell? Now remember, we're going to talk about this in great detail in just a little bit later in the semester, but for now, do note that it's all about genes contained in our DNA. These genes code for sequences of a protein, and these proteins are what give us our traits. The coded information in the DNA molecule is in the form of a messenger RNA. The messenger RNA carries the order to build our protein. Remember, when we're talking proteins, we're simply talking about polypeptides. And polypeptides are those long strings of amino acids. Moving on in our tour of the cell, we can look at number 3, the manufacturing and distribution center, which is the endomembrane system. Now, we've already talked about the word membrane, right? The phospholipid bilayer is what makes up all membranes that we find in every single cell. The endomembrane system, endo means within, so this is a series of membranes that are found inside of our cell. The first component of the endomembrane system is the endoplasmic reticulum. And previously I defined this as uppercase E, uppercase R. That's your shorthand you can use when you refer to this region. We think of the endoplasmic reticulum as the factory assembly line. In our factory, this is where we're going to put together important products that are used throughout the cell. The endoplasmic reticulum is composed of 2 parts, the rough ER and the smooth ER. The rough ER produces membrane proteins and new membranes, and the smooth ER produces lipids, including steroids. Now notice the rough appears rough because it is covered with ribosomes, and the smooth lacks these ribosomes, which is why it appears smooth. We know that ribosomes play a role in protein synthesis, so it makes sense that the rough ER would produce membrane proteins. The Golgi apparatus is another region, and again, we're talking about the endomembrane system, so this is just this continuous series of membranes folded in on each other inside the cell. And the Golgi apparatus is one of these folded regions of membranes that is going to allow for the cell to refine, store, and distribute the chemical products required for our cell. We can think of the Golgi apparatus as being the packaging center. It packages up those products made from the factory and gets them ready to transport. How do we transport things around? In transport vesicles. Here we can see is a transport vesicle that's pinching off of the membrane and carrying the products or the packages that were made by the factory. Lysosomes are another component of the endomembrane system because they form as little sacs that pinch off of the larger membranes. However, these sacs are important because they contain special enzymes called hydrolytic enzymes. We can think of lysosomes as being the maintenance crew or the janitorial staff. They're going to clean up the old, dead, damaged parts, but they're also going to help digest food and other types of molecules that are beneficial for our cell. Here's a human disease that is caused when your lysosomes malfunction, and that disease is called Tay-Sachs disease. It was first discovered over 100 years ago, and the telltale sign is that the doctor looks in your eye with a high-powered lens and light, and they see a red dot in the center of your eyeball. And unfortunately, this disease often comes around in children. That is to say, it develops in children, and it is usually fatal, because if you can't process the waste in your cells, you're going to be very sick. How does an enzyme get into a lysosome? Well, remember the endoplasmic reticulum? The rough ER is covered with those ribosomes, and ribosomes play a role in protein synthesis. So here we can see on this diagram the ribosome is making a growing polypeptide. The polypeptide folds into a three-dimensional protein, and then it pinches off in a transport vesicle. Now this vesicle with a hydrolytic enzyme is a lysosome. Another component of the endomembrane system are vacuoles. And these vacuoles are found in only some types of organisms. For example, a contractile vacuole is just found in some members of the Protista. Here's an example of a protist that uses these vacuoles for primitive movement. Another type of vacuole is important to plants, and it's called the central vacuole, or we also call it the water vacuole, because it fills with water and it's where most plant cells store most of their water. All right, our next stop in our tour of the cell is all about energy conversion, the so-called power plant of our cell, which is either chloroplasts and or the mitochondria. Mitochondria are the power plants for all eukaryotic cells. Chloroplasts are only found in those photosynthetic eukaryotic cells. Both of them are unique because these organelles contain their own DNA. And this DNA is slightly different from the DNA found in the nucleus in the cell. One explanation for this is a theory called endosymbiosis, and you can see that word on your screen here. Endosymbiosis tells us that these organelles evolved from free-living prokaryotes. That is to say, an ancestral host cell over time, which is to say billions of years, took in these ancestral photosynthetic prokaryotic cells and these ancestral aerobic heterotrophic prokaryotic cells, and over time included the directions in its genetic code to build these organelles. And we're going to talk more about endosymbiosis when we do our microorganisms lecture later on in the semester. Chloroplasts are those double membrane-bound organelles that are the site of photosynthesis. In our diagram here, we can see the membranes, inner and outer, and then inside the chloroplast are these little stacks of green discs. The little stack of green discs is called the granum, and then the fluid between the green discs is called the stroma. Both of these play a very important role in photosynthesis, which recall photosynthesis converts light energy to chemical energy, and the product, or the very important main product of photosynthesis, is the creation of sugar molecules. We're going to talk all things photosynthesis in an entire lecture devoted to this important metabolic reaction. Mitochondria are found in all eukaryotic cells, animals, plants, all eukaryotic cells. They are the site of the process called cellular respiration, which takes those sugars that were ultimately created by photosynthesis and breaks them down into a usable energy source called ATP. And that's represented by uppercase A, uppercase T, and uppercase P. And we're going to learn all about this ATP in another chapter coming up very soon this semester. So this is a challenging question, but let's see if you can figure this out. Which of the following is true of plant cells? The first choice, plants use chloroplasts to perform all the functions of animal cells. The next choice is plant cells use energy from sunlight to break down carbohydrates to drive photosynthesis. The next choice is plant cells use mitochondria for converting energy stored in carbohydrates. The next choice is plant cells use photosynthesis to produce all the energy that is needed to drive cell process. Or the last choice, all of the above are true. So that's tricky. When we have the all of the above is true, it makes us really think about all of those answers. So please pause the lecture now if you don't want to see the answer. And the answer is plant cells use mitochondria for converting energy stored in carbohydrates. And those carbohydrates are usually glucose molecules, which are types of sugars. All of the other choices here are not true. In our next tour of the cell, we're going to look at how cells maintain their shape and in some cases move, which is called the cytoskeleton. The cytoskeleton is a network of protein fibers, and it contributes two important functions. Number one, it contributes mechanical support, and this can allow the cell to maintain shape. Number two is it can change the shape of our cell in some types of organisms, such as this single-celled organism called the amoeba, and that allows for primitive movement. In either case, the cytoskeleton is very similar to the support beams of our factory. It holds together that factory and provides space for all of the important reactions to occur. The animal cytoskeleton has three major components: microfilaments, intermediate filaments, and microtubules. All three of these are very important parts of the animal skeleton. Our final stop in our tour of the cell is all about cell movement, and that is through parts called cilia and or flagella. Flagella allow for different types of cells to move forward by whipping that tail around. These are pretty rare in a lot of organisms. For example, in humans, we only have one type of cell that has a flagellum, and that is the human sperm cell. Cilia are a bit more common. These look like little hairs covering the surface of the cells, and these cilia move back and forth in a coordinated motion. For some organisms, like this protist, these cilia allow it to move and float through the water. But in other organisms, like humans, we have cilia that are lining the surface of our respiratory tract cells. Now that we've learned all the parts of the cells, it's important to understand that these parts don't work independently, but they are all interconnected. Cells stick together and communicate. Just as organelles in a cell work together, the cells that make up multicellular organisms also divide the labor, which requires communication among neighboring cells. Plants communicate through plasmodesmata, which are those little tunnels that are found in the cell walls that surround each and every cell. Animals have other types of ways to stick together. The first is called a gap junction. Gap junctions connect animal cells, and they are analogous to plasmodesmata, meaning they have a similar structure. They almost look like little tunnels that connect these animal cell membranes together. Tight junctions fuse the membranes of adjacent animal cells together, preventing substances from flowing between those cells. So the tight junction ties them very close together and allows for molecules to not move very efficiently. And then anchoring junctions, those are unique because those tie cells together, but it allows there to be some space between them. So it's not like a tight junction where they're very closely touching, but it's also not like a gap junction where it's a tunnel. So these are three different ways in which animal cells can stick together and communicate. Here's another question for us before we wrap up our lecture. How many of these organelles occur in our human skin cells? The choices are mitochondria, ribosomes, nucleus, lysosomes, chloroplasts, vacuole, and endoplasmic reticulum. Pause the lecture if you don't want to see the answer. The answer is 5. Human skin cells have all of the following except chloroplasts and vacuole. Consider this review question when you're studying for the exam. A cell has many molecules and structures, including the following: proteins, DNA, ribosomes, plasma membrane, cell wall, and chloroplast. This could be a cell from which type of organism? Make sure that you can explain your reasoning and provide evidence from this lecture and your textbook. I encourage you all to check out this interactive cell animation. By putting this link into your computer's browser, it's a very fun tutorial that shows you all the parts of a three-dimensional cell. A final word about cells is that all cells are not created equal. In general, we talk about plant cells and animal cells like they are these static things. that have very specific functions. But in reality, multicellular individuals have a huge variety of cell types found throughout their tissues and organs. For example, muscle cells look and behave very differently from neurons, and leaf cells have chloroplasts, but underground plant tissues such as roots do not. So this is just a couple examples about how there are a huge diversity of cells that are found in organisms.